Thanks everyone for coming tonight. It's been a while. It's nice to be back, Primary Health Network. So I'll be talking about the COVID vaccines tonight, and I'll be talking about a series of the candidate vaccines. But I think it's important, given the things that surround this particular area, the degree of interest and the degree of I guess, things that are still either dynamic or unclear. I thought I'd start by setting out a few things. So my talk, what is it? I'm going to overview the primary COVID-19 vaccine candidates with a focus on those that are currently part of the Commonwealth's deployment strategy. I'll go through what the mechanisms of action are, so how they work, what the available data is surrounding their efficacy and select senses, some of their adverse effects, and also go through some of their unanswered questions, things that we still don't know or are yet to be formally published about the vaccine candidates. What it's not is an attempt to analyze or roll out or describe the vaccination rollout strategy. That's not my area of expertise. It's not an area over which I have any influence, but it's also not an opportunity to kind of speculate about what policy changes or what cultural changes might come about once the vaccine rollout is well underway or when it's finished. Because, you know, I just work here, I don't make those decisions. And so I'm going to stick to kind of what I know best, which is the sort of vaccinology and the science that underpins it, recognizing that there are certainly world experts separate to that. I think it's worth spending a moment just reflecting on where we've come from and where we are. So these are some headlines from less than 12 months ago, really. And I remember giving my first COVID talk to this audience in this room less than 12 months ago. And I remember at that stage, the vaccines were that great white hope, something that was off in the distance, far away, something that we would all hoped to come, but something that we really couldn't use to justify any dropping off in our behaviours or any changes to how we managed all the new things we'd learned around concepts like social distancing, the new rigours we approached hand hygiene and infection control with. The vaccines really have been a remarkable achievement, and I think it's worth reflecting on exactly how quicker than anybody could really have anticipated, they've managed to come to the fore. Anthony Fauci said early in the pandemic that he was confident there'd be a vaccine within 18 months, and people said he was crazy. They said it was unsafe. But as a lot of other people said at the time, myself included, well, if anybody knows what the score is, it's going to be him. So up until now, there are 250 COVID vaccines in development, and the approval of the vaccine candidates we're going to talk about tonight is not going to slow that work down. There's always going to be things they could do better or a new modality or methodology to explore, and a lot of those works are going to continue over the next 12 to 18 months. There are a series of phase 3 studies still in recruitment, and they'll report gradually, but it is worth taking some time now that we've got the first few major candidates off the rank to look at what they can offer. So 75 of those 250 are currently in the clinical phase. So only the Pfizer biotech mRNA vaccine and the AstraZeneca Oxford vaccine are currently approved by the TGA. The third vaccine partner that I'll talk about in detail tonight is Novavax. The government has ordered 51 million doses of Novavax, even though it's not yet approved by the TGA, and its phase 3 trial is purely an interim analysis. It's not formally been published as far as I'm aware. But that, and I'll talk about why those kinds of things take place where the government orders a vaccine that isn't entirely approved or published in a second. There are a series of other vaccine candidates around the world in various stages of deployment, and these are some of the major ones. The Mrna 1273 vaccines and mRNA vaccines similar to Pfizer, and the efficacy studies that they've published show a pretty similar level of activity around efficacy and antibody generation. Already mentioned Novavax, which is a virus-like particle or a recombinant viral particle vaccine. Johnson & Johnson vaccine is currently also in trial format in a number of countries around the world. That's an adjuvant vaccine. And Gamaleya, or the Russian Sputnik 5 vaccine, I'll talk about it in some detail because it's a really interesting story, actually, really a few interesting questions about that vaccine. And that's currently in rollout in Russia, but a number of other countries, including Argentina and Mexico. There's also a couple of other vaccines currently in use in China, which have some novel mechanisms of action, but I've not listed them because they're not widely in use outside of the Chinese setting. So to go over the breadth of the vaccine candidates, again, this is some data from WHO's Vaccine Candidate Register. So the World Health Organization keeps a list of all the vaccine candidates currently under development and updates it regularly as they progress through their trial phase or they're ceased. So you can see that the dosing schedules that the Australian vaccines use, so a 2-dose schedule is by far the most common. By far the most common route is injectable, usually intramuscular. And it's pretty typical for vaccines, and we'll talk about how those regimens come about in some brief detail. But certainly there's 14% that are to be determined, or for which there's no data. I know at least some of those are intranasal, because the idea of intranasal vaccines is that it primes the local and innate immune systems in the upper airway where the virus is first exposed to, and may confer a higher degree of immunity theoretically. But none of those vaccine candidates have progressed far enough along to really consider them a viable option at this stage. So I talked about how quickly we've managed to make the journey from discovering COVID, adapting to the acute threat, to having a vaccine rollout. And I know from speaking to a lot of people in my day-to-day practice that that's something that people greet with both a large amount of interest, or a large amount of positivity, but also there's a certain subset of people who feel that that's something to be skeptical about, this idea that it happens so quickly, there must be a problem in the process. But I think it's really important to go through how vaccines traditionally arise and some of the market forces that maybe slow them down along the way that we were able to put to the side here with COVID. So, typically the traditional approach is that you work out what you want to vaccinate for, you study it, you create some novel approaches, some ideas that might be not previously in use. The amount of resourcing dedicated to that is small. Grants progress slowly. You've got to win funding. You've got to put through your ethics. You've got to, justify its existence. And then you've got to progress it through each of those phases, phase 1, phase 2. At every stage, you've got to convince the mechanisms of healthcare and governance that it's worth continuing and that it's safe to continue. And then eventually you scale up to a manufacturing level, it's licensed. And at every stage, there's this question of, well, not just, is this good work, but is this good work that's viable, that's going to keep going, it's going to deliver a return on investment. In the outbreak setting, a lot of those market barriers go to the side, and you've got a huge amount of resource and a huge amount of man hours and woman hours, and a huge amount of dedication that goes into those things. So you'd be surprised, and if anyone reflects on the kind of changes that you made to your work life or your home life for COVID, you can kind of get an analogous approach, right? So the changes that we made in the hospital setting where I work to something as simple as the visitor policy, under normal business, that would take 6 to 12 months to get done. But in COVID, in an outbreak setting, we were making changes on the day, right? And that's because there's urgency, there's buy-in, and there's for the vaccine paradigm, there's market forces simply aren't necessary. And actually, the same market forces that are usually a barrier actually worked advantageously when we were talking about the development of the COVID vaccine. So these are two sort of title headlines from papers that were published well before COVID even became a word anybody understood. These are papers from Vaccine above, and below I think it was Research Microbiology, that demonstrated that vaccines for both the original SARS-CoV virus and the MERS virus were viable, were safe, and there was a reasonable chance of success should they be deployed down further lines. But the reason that vaccine, you may have never heard of the SARS vaccine or the MERS vaccine, is that the commercial viabilities of these products were never apparent. They were never viable. So all this work was done, but no one could pay for the second phase. There is no market for a vaccine against MERS, a disease that has functionally been eliminated outside of very small pockets. There is no the market for a vaccine for SARS. And so the large trials just simply never came to fruition. They were never necessary as far as the huge amount of cost that goes into generating a vaccine and bringing it to market. But this work was not for naught, because this is the SARS-CoV-2 virus, or the virus that causes COVID. And you can see on the right-hand side, there's an electron micrograph of the spike protein. So the spike protein actually exists in an analogous form in both the MERS and the SARS-1 virus. And the work that these researchers did in identifying it as a key target for vaccination, a key target in antigenic stimulation and the immune response, and developing particles and vaccine methodologies that worked for them, actually gave the researchers working on the SARS-CoV-2 virus a heck of a head start. And so the spike protein that they manufactured those antibodies, those vaccines against, in the initial waves of those research, some of that research is over 10 years old, formed a very clear platform for all the work that they're doing now, because all the vaccine candidates approved in Australia are manufactured against the spike protein. Okay, so you can see a very clear pathway and a very clear way by which we managed to get from work done to work completed, even on the COVID-19 vaccine, even though we've only really been able to sequence the genome of this particular virus for approximately 12 months. So talk about some of the vaccine platforms. I mentioned the ones that we have available in Australia. There are some traditional vaccine platforms that we use very frequently already. So you can have inactivated or attenuated viruses. So inactivated viruses are viruses that were live. They're inactivated through chemical means and then injected in order to prime the immune system. There's actually a couple of the Chinese vaccine candidates that are currently in use use this methodology, but none of the Australian ones. So you can have live attenuated virus, which is similar, but it's virus that's alive, but is altered in order to render it safe and non-infectious to the host. These are the kind of viruses you have to be mindful of when you're giving vaccination to the immunocompromised. So measles being the sort of the archetypal one, or the varicella vaccine, are both live attenuated viruses. There are some in preclinical studies for COVID, and this actually is considered probably the most promising route for developing an inhaled vaccine, which is more immunogenic, but because it's the model they use for the live attenuated influenza vaccine, inhaled method, but we're not far enough across, as I said. And recombinant vaccines, so vaccines that rely on expressing viral particles in a non-human cell, and then using that as kind of a vehicle for the exposure of the immune system to a viral antigen. And we'll talk about the Novavax vaccine, which uses a similar kind of approach, but rather than using virus particles, uses a virus-like particle, a subset of the whole viral structure. So vector-based vaccines are important because it's the basis for the vaccine platform for both the AstraZeneca or the ChAdOx and the Sputnik vaccines. So vector-based vaccines can be either replication competent, so they can actively replicate within the host, or replication incompetent. And basically what that refers to is the viral vector. So they use a non-COVID virus, and they design it to express elements of the COVID virus on its surface, and then use that as an entry point or a kind of a vehicle to expose the body to the COVID-19 antigens without having to expose live or infectious virus. So I'll talk a little bit about some of the efficacy data. So this is from the Lancet paper that covered the ChAdOx NCOV1 vaccine. So this is the AstraZeneca vaccine, and this is some pooled data between both the British and non-British arms of the trial. And so you can see here clearly that there's that headline number of a vaccine efficacy of 70%, sorry, there on the far right. But you can see there, I've read that mostly to show you the numbers. So there's about over 10,000 people in the trial in total. And the event rate, which is symptomatic COVID, and I'll go into how they define that in just a second, is very clearly 30 in the treatment group, which is we give them vaccine, and 100 in the control arm. And then you'll see in this table, it mentions the LD, SD, and the SD recipients. So what that refers to is you may have seen some questions about a low dose and normal dose protocol. So the guys who were running the trial in the United Kingdom pivoted their trial structure to go from a low dose of vaccine followed by a standard dose to a dual standard dose once they got some interim data to look at the immunogenicity. So you've got those two groups mixed together, and I don't think there was enough, particularly in this group, in this vaccine, there was enough data to really make a statement about which was better, because the numbers, as you can see on a couple of slides over, are pretty comparable. And so this is a point that I want to make on this slide is about endpoints. So you look at this idea of on this slide about vaccine efficacy. Well, what is an efficacious vaccine, right? Like, what is it? What do we actually measure? You know, do we measure who gets hospitalized? Do we measure who's infectious? Do we measure, basically in this trial setting, the answer to that question is cases were defined by people who rang up the trial management group and said, I have symptoms, I think I need a test, as they were asked to do. They were checked in on regularly to ask the question. Then they went in, got a test, and got done. So in those settings, you won't be able to capture people who had asymptomatic transmission, but you're able to, your case numbers define people who had symptomatic COVID. But that's everything from a fever to a cough to a runny nose. But it's important to note that in the Oxford AstraZeneca trial, out of those 12,000 people who were vaccinated with the candidate vaccine, no one got severe COVID. So no one got severe COVID less than 21 days after. No one got severe COVID more than 21 days after. No one got hospitalized except for in the first 21 days after the first dose, so before the booster dose was given. And that's important because when you're thinking about what you want your vaccine to do, it's about more than ever preventing you from getting the infection. A vaccine that ameliorates severe disease and prevents anyone from even being hospitalized for COVID-19, irregardless of their age or comorbid profile, would be an excellent vaccine candidate, right? And so you've got to keep your focus on what we're trying to achieve here. And certainly the AstraZeneca vaccine, from all the available data here and experience overseas, does a very good job of mitigating severe disease. It may not necessarily ameliorate any symptomatic COVID, but no vaccine candidate we have access to does that at all. So you can see here the breakdown in ages. So in the low dose, standard dose protocol, everyone was young, and you can see those numbers there. And the control group were given the meningococcal ACWY vaccine. So that's in order to kind of, they don't give you saline, they give you MenC, so you have some degree of ability to, you can't infer necessarily from the fact you got a fever that you must have got the proper vaccine, or a bit of pain or a bit of myalgia after the event. And so you can see there that there's, when they entered into the standard dose, they had both a UK arm and a Brazilian arm, and they had a cohort of people over the age of 70 made up about 10%. Certainly the safety data from those groups there in the advanced age group was not, it was, you know, didn't give you any signal of an increased rate of adverse event, but it did, it's important to note because you include people who are over the age of 70 in that group of people who weren't ever hospitalized. And those are the kind of patients who are most likely to land in hospital with COVID-19 infection. So demonstrating that you can protect this group, the over 70s, is a really important part of how you judge the efficacy or the usefulness of this vaccine candidate. So you might have seen some other things rather than around the dosing strength. You might have seen a bit about dosing interval, and this actually is really important and really interesting. So when AstraZeneca first marketed their candidate vaccine, they specified a period of time between the first dose and the second dose, and that was about 4 weeks. But what they found in the phase 3 trial, so in the trial managers actually took over and did the management, they found that the longer you waited between your first dose and your second dose, the more robust your immune response became and the more efficacious it appears from the data the vaccine was. And that's why you might have seen there's a delineation between what the World Health Organization and the UK government recommend in terms of how long you wait between doses of the AstraZeneca vaccine and what the manufacturer recommends you wait between 2 doses of the AstraZeneca vaccine. And this is just to illustrate that even though the vaccine efficacy goes up as you wait over time, the degree of protection waiting since the first dose doesn't drop. So you can see that after 1 dose, so this is symptomatic COVID-19 cases after 21 days after a single dose, whoops, and because your main concern would be is that if you waited between doses, you'd increase someone's risk of getting COVID between the doses you wait. You have to, between the dosing interval, but there wasn't a significant drop off in the efficacy of the vaccine between the 22 and 30 day mark, or 22 and 90 day mark, I'm sorry. And so this is some data moving on to some interim results on vaccine efficacy. Whoops, where am I going here? Sorry. This is some data out of I think this is out of the Pfizer group. So this is, yeah, where am I now? Sorry, I've just lost the position of my slides, I'm sorry. Symptomatic COVID-19 cases. Oh yeah, no, this is some more data, sorry, out of the AstraZeneca vaccine, just looking at the spread of age groups and to demonstrate its efficacy in those groups. You can see the spread of age groups across there, and you can get into zero, moderate or severe COVID cases in the vaccine group. I'll move here. So I want to talk a bit about the Pfizer vaccine. And so the Pfizer vaccine is a really interesting vaccine platform because it's an mRNA vaccine. And so what's really interesting about an mRNA vaccine is that it's really the first licensed mRNA vaccine we've ever seen. And so what I mean by that is, when I say mRNA vaccine, is that I mentioned all the platforms I used earlier required a viral carrier or a cell carrier to bring it in, but actually what the mRNA vaccine does is it basically custom designed and gene edited a messenger RNA coated in a lipid nanoparticle. And what the lipid nanoparticle does is it prevents degradation. So it's basically, again, it forms a delivery system, but it's rather than being a virus of another kind or a cell of another kind, it's just custom designed nanolipid layer. So that what happens is that mRNA, which is designed and manufactured really, really rapidly through the use of viral plasmids in bacterial cell lines, is able to be produced and then introduced into the host cells. So these are the cells of the vaccinated person. The mRNA is recognized by the cell's internal factory and internal mechanisms and is translated and processed. And then viral antigens are then immediately produced and expressed on cells to allow for immune priming. So basically what it is you're telling your body's cells to make a tiny bit of virus using the messenger RNA, and then that tiny bit of virus is expressed on your own cells and then helps to prime your immune system to recognize the virus. Really interesting form of vaccine technology, and one that is really important because once you have the infrastructure to start making them, which we don't have currently in Australia but are exploring, you can manufacture huge amounts of virus of mRNA really quickly, because what the typical limiting factor for most traditional vaccine productions is you have to grow virus. And anybody who's works in testing for virus frequently knows that viral culture is not a test we do frequently in the community, and it's because it's really hard to do. Growing viruses in a laboratory setting is extremely difficult and slow. And so being able to bypass that by simply using bacterial cultures that produce plasmid mRNAs that we can pump out at speed really will mean the amount of vaccine we can produce once the infrastructure exists is extremely high. It's one of the reasons why the amount of mRNA vaccines that have been distributed across the larger population centers of the world is so significant. And so people, I guess, have equally expressed some concern that, well, because it's a new vaccine type, it must be somehow less tested or less kind of experienced. And so this is just a slide to demonstrate that although this is the first vaccine using the mRNA structures to be brought to market, It's not the first one to ever be invented or used or demonstrated. The same market forces that I mentioned applies to all vaccines, apply to this vaccine technology. And so the work on demonstrating the safety of these vaccines and the efficacy of these vaccines in producing an immune response was already done. It was simply a matter of pivoting that work to COVID and allowing that COVID work to be capitalized on and become the first licensed candidate. But certainly we've been working with mRNA vaccines for some time. This is some data out of the huge, it's really remarkable how much data there is here in the Israeli study of the Pfizer vaccine. So the Pfizer vaccine has been rolled out across the world in really significant numbers, and one of the major centers that really accelerated their deployment was Israel. And so this is some data to show in a study of almost 1.2 million Israeli citizens are part of this study, and it basically demonstrated that the Pfizer vaccine was extremely effective in reducing symptomatic COVID-19, but also, as I mentioned, similar to the Oxford vaccine, hospitalization and death. And so you can see those numbers, the Kaplan-Meier curves there are all extremely convincing. You can see that there's a lead time where after the vaccination, there is some ongoing events in the vaccinated population as the immune response is still being primed, but certainly you can see once they hit about that 20-day mark, they really start to separate quite markedly. This Israeli study was great because it's, again, similarly to the elements of the AstraZeneca vaccine study, they used a really great cross-section of people. So you can see now that the median age was 45, but they had a very high number overall in terms of absolute numbers of people above the age of 70. So, you've got about 80,000 people there who were all given the vaccine over the age of 70, and we didn't see a significant safety signal, but we still saw an efficacious response when it came to limiting the amount of severe disease. Because that's, you mentioned it, we'll mention it when we go to looking at some other studies, particularly looking at the variants, that if you don't study a population that's at risk for severe disease, your ability to demonstrate a presence or absence of severe disease is meaningless. The trial will be underpowered. So everyone wants to talk about adverse reactions when it comes to the Pfizer vaccine. What's clear is that this mRNA vaccine and the Moderna one just like it are extremely immunogenic. What that means is that they produce a robust immune response, and with that robust immune response comes side effects. Now, I want to just spend some time laboring the point about exactly what we're talking about when we talk about side effects and what we can expect when we might be caring for people who've received it, or we might be receiving it ourselves if that is the candidate vaccine for which you are eligible. So BNT162b2 is the candidate vaccine, or the Pfizer vaccine. This is taken out of the FDA approval paperwork, so it's admitted as part of its initial safety application. So you can see that depending on, irregardless of what dose we're talking about, the first or the second, the amount of local reaction was essentially the same. So about a quarter of people had moderate amount of pain at the injection site in both groups, and they had, much smaller numbers had redness or swelling. But what's interesting is that the more systemic effects, they tend to be, there's clearly a priming effect between the two doses. So when you look at fever, any fever above 38 degrees, only 3.7% of people had it at their first dose, which is pretty par for the course, really, when you're looking at immunogenic vaccines. But that number, interestingly, rises to as high as 15% on the second dose. So there's clearly a sense of, and this makes sense, right? Because you use the first dose, the whole concept of the first dose is to prime the immune system and then boost it with the second dose. You've already got a baseline immune response by the time you have your second dose. So it makes sense that you'd have a more robust amount of systemic side effect when you reach that point. And you see that, but the numbers of higher grade fever, so above 39 degrees, becomes very rare. Fatigue of any kind is reported at high rates. Moderate fatigue, particularly at the second dose, is up to almost a third of people. Headache, it's common to any degree, particularly mild to moderate headaches, again, happen about a quarter of the time. Chills are common. So you can see this is like, for a while after your second dose, you've got a pretty high rate of being relatively SIRSy, right? Like feeling a bit flat, being a bit myalgic, having flu-like symptoms. And actually, this is something that comes up a lot when we're talking about the deployment of the Pfizer vaccine in the healthcare setting. So, when we've got 1A staff members in Orange Hospital, we talk about, well, what are we going to do if we vaccinate our entire ICU workforce in one day, and then half of them can't come to work the next day because they've all got systemic symptoms and the kind of symptoms that we've been telling people to stay away from work with because they might have COVID. And the vaccine trials are very clear that everyone who suffered these adverse effects was tested for COVID because they met criteria, and we can't be confident that flu-like illness at day 2 after vaccination is definitely caused by the vaccine. This is the third candidate vaccine that I wanted to talk about. This is the Novavax vaccine, which again is really interesting pathway. You can kind of read it for yourself there, but basically it's a recombinant vaccine candidate where it takes a virus-like particle, so the spike protein, so not the whole virus, just part of it, and it puts it inside a baculovirus, which was not a virus I'd heard of before I read about this, on its cell surface, and then it uses the baculovirus to induce the DNA into a moth cell, and then it takes the spike proteins that are made by the moth cell and puts them on a synthetic particle as like a stud kind of thing. It kind of builds a honeycomb using these spike proteins made in the moth cell, and then it adds it with some saponin, which is there as an immune primer, and then injects it into the vaccinated person, and that allows the immune response to prime against the spike protein, which, as I said earlier, is a key part of all the immunogenicity of our vaccines, and is one of the major components that when we test the convalescent sera of people who've recovered from COVID, that's where their antibodies are made against. So Novavax is interesting in so much as that they're a great example of what really has generated a lot of behaviors within the COVID world, which is that traditionally, you wanted something out there, you'd submit your data, it'd go through peer review, it'd get, to be delayed. Like, it's not uncommon to wait 12 months between having an article accepted and having it published. And then because of the speed we're working at COVID, we went from, we can't wait for all the articles to be published. We've got to do preprints. And so preprints were these things where, they're not peer reviewed, but they're, the journal recognizes it's of such significance and importance, it already publishes it while peer review is in process. And then companies decided that wasn't enough. We need to get the message out even faster. So they basically just started doing medicine by press release. And so this is from Novavax's phase 1 study, which is really all that's published at the moment. That was actually performed in Australia out of Melbourne and Brisbane. And basically the example of this is just to demonstrate that their candidate vaccine is immunogenic, so it produces a significant amount of antibody, and that amount of antibody is targeted, as I said, at the spike protein, and it's comparable to the convalescent sera of patients who've had COVID. So it kind of produces a degree of antibody comparable to someone with COVID. And that's always the caveat that when we talk about vaccine studies is it's not necessarily ideal to infer linearly between the amount of antibody you produce and your degree of protection. That's been a problem we've had with influenza vaccines for ages, that a low level antibody doesn't mean you're not immune, and a high level antibody doesn't mean that you definitely are. But it's the best marker we have in phase 1-2 studies before we get into the real world and start seeing how many infections it's actually able to prevent. The Novavax guys have released some interim data from their phase 3 studies, which says it's got an efficacy of about, I think 87 to 88%. That data is yet to be peer reviewed, so not even be published in peer review form. So I haven't laboured the point because I think that's a bit preliminary, but certainly the government has invested some funds in securing these doses because they're slightly easier to store than the Pfizer vaccine, and the cold chain is probably something we've heard a lot about with the Mrna vaccines, and it's what the recombinant vaccines are slightly better at being able to manage that variation in temperature. So everyone wants to talk about the variants. The variants are, they're a significant issue, but there's a clear delineation between the two major variants of concern that you might hear about. So the first is B117, which is the UK strain, so the major endemic variant now in So it is likely to be more transmissible and significantly more so. I mean, all you had to do is look at what happened in Brisbane and Melbourne. You can see that, there's both a policy response, but also a clinical evidence that says that these things move extremely quickly. But the good news is that the United Kingdom is currently vaccinating with AstraZeneca, and they've not seen a impairment to their case number improvement in areas where the variant is high. And they've also done tests to look at the preserved antibody response, and they've found that all of AstraZeneca, Pfizer, and Novavax seem to show a preserved antibody response and a preserved degree of protection when dealing with the B.1.1.7 variant. This is some data, again, out of the AstraZeneca study to show that when they look specifically at the B.1.1.7 variant, there is a significant amount of vaccine efficacy. So they were able to look at asymptomatic infections in this cohort, and you can see that asymptomatic infection, it wasn't particularly well powered for that, and the confidence intervals are wide. But you can see down here that it's likely that there is some degree of protection against the B.1.1.7 variant, maybe a minor impact, but not certainly a significant one, an overly significant one. I'm sorry, I should say. Now, the South African strain is a slightly more concerning variant for a couple of reasons. So basically, it's got a number of mutations that make it a worry, and the worst one is this E484K spike protein mutation. And so some of the therapeutics of COVID-19 focused on monoclonal antibodies directed against the spike protein. So they were very effective in wild type virus really early on. They were expensive and they made some difference. But in a laboratory setting, when you exposed virus that had an E484K mutation to those monoclonal antibodies, they were 40 to 60 times less likely to be effective in neutralizing the virus. And so the concern here is that it varies the spike protein enough that the existing immune response, both to people who've had COVID before and the people who've had that degree of immune stimulation induced by vaccine, is going to be significant. And it's certainly been demonstrated that, according to some early data that the guys at Novavax have released, that you can be reinfected with a B.1.351 variant. So a South African variant can bypass natural immunity and cause reinfection in a way that is sort of significant for we're talking about, natural herd immunity, I suppose, in those settings. Novavax, Mrna, and AstraZeneca are the companies that have currently released some testing into their, Pfizer has as well actually, released some data around the efficacy of their vaccine candidates against B.1.351. All of them show a muted antibody response. So certainly there is less antibody production in response to that variant. And that's what is unclear is what degree of impact does that have on vaccine efficacy? And because it's just not, that data takes a while to develop, and we're not really certain at that stage. This is some data out of the Pfizer group, just to demonstrate that Pfizer, the same as the other three, also demonstrates a decrease in the production of neutralizing antibody compared with wild type. So this is the neutralizing antibody produced in response to wild type virus. This is the neutralizing antibody produced in response to a B.1.3.1 virus. And you can see that cells that have been primed against the vaccine, primed against the antigens produced by the vaccine, have a much lower antibody response in response to a B.1.351. So I'll finish on the question of transmission. So what everybody, I guess, wants to know is, what does this data have to do with efficacy against transmission? And so what I guess the overwhelming view of the experts who have access to much more data than me and are much more experienced in this area is that it's highly likely that, given the efficacy of these vaccines in preventing symptomatic disease, and from what we can see, preventing asymptomatic disease, they're very likely to have a significant impact on transmission. And we'll see that the way to prove that is to demonstrate that in an epidemic setting, when you start vaccinating people, the case numbers drop off inordinately compared to the number of people you've vaccinated. And so we'll have to wait for that data to come through, but I think it is important to demonstrate that we don't have any hard data to say that one vaccine candidate is better at preventing onward transmission and is therefore better from a public health and herd immunity setting. And we certainly don't have any head-to-head data between the two vaccines, because each of those studies that I showed you were studies independent of one another. They were conducted in isolation. So their protocols around how they tested patients, where they tested patients, the settings in which they were conducted, how much COVID was around, you know, what degree of follow-up they had, what the study population was in terms of age. All of them are different. And so I guess one of the major take-home messages I'd like to get across here is that we might see headline numbers, 75% efficacy, 95% efficacy, 20% efficacy, but those numbers they're not comparable between one another. There's no scale to say that a vaccine that's 90% efficacious in one study is better than a vaccine that's 85% efficacious in another, because you're comparing apples with oranges. That data will become available and it will inform our decision making when it can, but it's important to, you know, give the science time to evolve, because that's what served us in good stead up till now. So that's the end of what I had to say. Pass over to my learned colleague.
We do have a couple of questions that are pertinent to you, Jake.
Oh, yeah.
Should we give the vaccine to pregnant women? It's a question from Dr. Watson.
Oh, it's a great question. So, Marty, I guess there's two arms to that answer. The first is, does the data say it's safe? The second is it a recommendation of the policy frameworks? I guess that's the thing, because when you deal with women in pregnancy, your safety barriers are much different. There were absolutely pregnant women in both the AstraZeneca and the Pfizer trial. There was about 1,000 pregnant women in the treatment, in the vaccine arm of the Israeli study, but there wasn't an increased safety mechanism. Certainly, certain overseas bodies have recommended that pregnant women get vaccinated. I think ultimately, in terms of will we in Australia have have an instruction, should we give vaccine X to a pregnant woman? I would wait for the policy frameworks, because the appetite for risk is something that the policy guys will work through based on the available data. But I don't think there's a large safety signal currently to say that we shouldn't.
Another question from Dr. Watson: is it likely to work in immunocompromised people, such as organ recipients of immunosuppressants?
Yes, it is. So there is, again, going backwards to that Israeli study, there is a cohort of people who have immunosuppression, and it was effective in that group. Because it's not because it's an mRNA vaccine, I'm talking specifically about Pfizer. Because it's an mRNA vaccine, it doesn't, your immunosuppression shouldn't necessarily mute that response to a great degree. I certainly haven't seen anything to say that any of the major vaccine candidates have been classed as not appropriate for the immunosuppressed.
Might move on to Tab's presentation now.
Thank you, Jake. That was really great. So just to give you a bit of context tonight, my job is to give you a really, really fast 10-minute whirlwind overview of what we're doing, I guess, getting ready for the rollout, knowing that it's fairly imminent. And I would just like to also start by acknowledging the people of the Wiradjuri Nation and the different nations across our area where people sit tonight, and welcome any Aboriginal and Torres Strait Islander Australians who are present in this meeting. And just acknowledge the artwork that's been developed for the COVID vaccine rollout for the Aboriginal community. And I thought it was a nice segue into what I'm going to talk about, that the artwork represents being connected to community and staying informed on the latest reputable health advice, and we become stronger as a whole community. And I think these are the two things that we really need to think about in our strategy as we go forward. Is we need to stay updated because it's changing every day, and we really need to work together as a whole community, because this is a monstrous task and it's really going to take all of us and all our resources to be able to roll this vaccine out across our region. So you may have seen our vaccine rollout strategy. So it's in phases. We're currently in phase 1A, or right at the very start of phase 1A. And then as we move through the calendar into March, we'll move into phase 1B. I think we're looking at phase 2 in about May, and these phases will intersect, so they won't be the beginning of one and the stopping of another. You're going to find that all of these phases will be overlapping as we get more vaccines, as we get easier to store vaccines, and we get different services coming on board able to vaccinate the population. Just for anyone who's not aware, for the start of this, there's vaccine hubs in Australia. There's 30 to 50, and our vaccination hub for the Pfizer vaccine is Dubbo. So vaccinations will be stored and dispatched out of Dubbo through New South Wales Health. So we're starting basically where the evidence tells us to start. So the government has a group, ATAGI, which is the technical advisory group, who provided information to them about what the evidence suggests the groups we should prioritise first. So you're probably quite familiar with phase 1A, but it's quarantine, border and frontline healthcare workers, which we realise is super essential right now. Our frontline healthcare workers, some of them have been prioritised, so we're looking at staff in hospital facilities, and there's a cohort of them, laboratory staff handling potentially infectious materials and taking samples. We're looking at our GP respiratory clinic staff, and we're also looking at aged care and disability care staff and aged care and disability residents. So this is the first place that the vaccination will start, and it's really starting ROCFs imminently. We'll then move into the phase 1B, and this is where we'll see our primary care providers start to be able to give vaccinations. So this week we've seen our general practices become notified about their EOIs. So we know that we have about 55 practices across our region who will be taking up the work of providing vaccinations. And we also have been notified about our respiratory clinics. We are still waiting on some further instruction around some of our other services, which I'll talk about shortly, but they have a brief to vaccinate our elderly aged over 80 years, our other healthcare workers, so most other healthcare workers who don't fit into our category 1A will be a 1B, so that we can keep protecting our medical staff. Adults with underlying medical conditions and disability, our critical high-risk workers, so we'll expand it to defence, police, fire and emergency services and meat workers, because we know that they're high risk. And I've just popped in there a little tool that you might be able to send people to if they're asking about their eligibility. There's actually an eligibility checker on the government's website, so it might save you some question time if you want to start to provide that to your staff and to other people. Just a little word on our vaccination rollout for our First Nation communities, and I've popped 2 resources in here for you. I would encourage you to listen to the mission, Dr. Jason Agostino, on the plans for the rollout through our Aboriginal medical services, but there's also some really wonderful advice for mainstream providers caring for Aboriginal patients about Aboriginal patients presenting for a vaccination, and the advice is that if they present and they identify, then they should be vaccinated. There shouldn't be a question, it should be accepted, and this is one of our national priorities. So have a listen, I've put the link in there, it's actually really good. And we've had over 100 of our AMSs and ARCHO services in Australia put their hand up to be vaccination sites. The outcome of that's still yet to be given to those sites, so we're waiting to find out what that is. But we also have a community engagement kit that you can start to share with your staff and share with your patients about how to engage the Aboriginal community in conversations around vaccination. And we know that this is a sensitive issue for some Aboriginal people because of the history of vaccination in Australia, so it's really a time to start thinking about your approach, your cultural safety around this, and having some resources for the Aboriginal community that you might want to promote the vaccination, because we know they're vulnerable, we know they're a high risk group, but we have to approach it the best way possible. So phase 2 and beyond, so once we hit about May, we will hit phase 2, and that's where we'll open up the criteria more widely. You'll see that the age begins to drop for adults and for our Aboriginal people right down to 18, and any other critical high-risk workers who haven't been vaccinated in phase 1. And then in 2B, we'll be looking at the balance of the adult population and catching up on our unvaccinated Australians from previous phases, which we know is going to be a challenging time for everybody. And then phase 3 is tipped as our under 16s, if and when that's recommended. And I'm not here to talk about the clinical guidelines around the vaccines, but there's more work to be done in that space. But we need to keep in mind that the younger age groups in our population make up about 25% of our whole population and about 40% of our Aboriginal population. So this could be a really key group in that strive to achieving herd immunity. And it also highlights the fact that it's important to be getting our adult population well vaccinated. because we still have this unknown about this age group. And community pharmacy will come on board in phase 2A, and I think that's going to be a welcome relief for our clinicians who will have now been vaccinating solidly for a few months. So you'll have your pharmacy colleagues on board to come along and help you to clean up some of the people who potentially don't attend services, and to run some campaigns about getting the wider community vaccinated. And we all know that our pharmacists are really great in this space. I was talking to a general practice today who's going to invite their pharmacists along to do some vaccinating with them in the earlier phases and make sure that they're confident in using multi-vials and just in the process. So I thought that's a really good example of working together, because we know that we're going to need to be doing these things in our small communities to ensure that we have plenty of vaccination services. So there was 4 EOIs, and this is where the complexity of the system comes in. As I mentioned, general practice EOIs, which we now have some clarity around. We also have the NATRO organisation looking after the AMS and ARTRO EMIs, which are still in the process of being determined. Our GP respiratory clinics directly via the Department of Health, and that's now been determined. Plus our pharmacies via the PPA, the Community Pharmacy Agreement Portal. So our pharmacies will be determined with the Pharmacy Guild. So as the PHN, we will be tracking all of this for you so that we can pull together where our vaccination sites are, who our vaccinators are, and have, I guess, a footprint for our community and our clinicians and for New South Wales Health, because we also need to identify if there's gaps in our region and if there's communities who don't have access to immunisations, and work out how we're going to get immunisations to those communities who might be smaller and potentially more vulnerable. So really, I think in summary, our vaccination rollout is going to have to be a partnership approach, where as a region we know that we're always service poor, and we cobble all these people together, all these services together, all these phases together, and really work to get a good outcome for people vaccinated in the region. Within the PHN, I just wanted to also mention that we have a tool that will help us to track this. It's called Capacity Tracker, and you should have all received some kind of information about this. The benefit of this is it will show us, and you can see a little map, it'll show us where our vaccinators actually are. We can share that with other healthcare professionals. We can track the rates of vaccination in our aged care facilities and our aged care facility staff, which is super important. You can alert us of issues if you can no longer vaccinate for any reason, whether it be a technical issue, a staffing issue, a vaccine supply issue. We can be alerted straight away and start to work with you on mitigating that. And we can also look where vaccines are in our region. So if we have oversupply and undersupply, not really knowing exactly how many of these things we're going to have, we can work to make sure that we are making it that we are covering as much of our region with supply that we can. So if you don't know about this, feel free to contact the PHN and we can help you set it up. Just a quick slide, and I won't go through it all, but we have had a lot of questions about getting practices ready, getting services ready. And look, there's plenty of things that you can be doing now. You can be looking at your site, looking at your accreditation standards. Check out the national booking system, which the government's utilising hot docs, and there's some information, early information about on that and how that might help you streamline your appointments and bookings. You need to be on the AIR so that everything goes up to the immunisation register. Maintaining your cold chain and having contingency plans if that goes down. Looking at your workforce and whether you need to surge them up for this and train them up. Your consumables, thinking about up-to-date CPR and emergency management processes, how your clinic's going to flow. You need to be all over adverse event reporting to the TAG and the public health unit should things happen. Looking at your communications with your patient and that local networking. Who else in my community do I need to tell that I'm immunising, and who else is immunising with me? One important thing that I've had heaps of questions over the last few days is staff getting ready for the COVID vaccination. They must have done the COVID vaccination training module, which is free and online. So you can start doing that now with your GPs and your nursing staff so that your team is actually ready. Rumour has it takes about 4 hours, so don't leave it to the night before.
Definitely don't skip.
And just from a PHN perspective, we're all here to support you through this, all the usual suspects. So I'll share these slides and you'll have all our contact details around any of these things. If we can help you get started, if we can help you get set up, if you need assistance, ask questions, walk through it with you. We're happy to help. And the Public Health Unit has also made the kind offer to support us if we have logistical questions around setting up immunisation sites. So there's plenty of support available. And my last point is just, we need to keep calm. We need to keep focused on the long game, because this is an unprecedented vaccinating situation that we've not been in before. And we need to learn as we go and adapt, because the evidence and the information continues to change. So keep calm and play the long game. And as I said, if you need any assistance, contact the PHN. I thank you for your attendance tonight. I know night times are always a push when we're busy, but we will send this out, and I've included all the resources that we've been, I guess, promoting for practices, so you can start to put together your plan for your practice and tick your boxes and get ready for March. Thank you.
Thanks, Tab. We do have a few questions that have come through, and we can start with an email question from Narelle. She works in a residential aged care facility and has said a resident spoke to her about previous anaphylaxis after taking acetazolamine and or sulfur, and was concerned with the risk.
Yep. So it's important to know that like all vaccines, the COVID vaccines will contain co-formulants. So things that are required to keep them stable or keep them active or able to tolerate the low temperatures, despite as well as the active components. The only major contraindication is if one of your patients has an anaphylaxis to one of those components. Those components are rare. None of the things that you mentioned strike me as parts of either any of the candidate vaccines, but it will be part of the product information, the dispensing information, what those components will be.
And maybe a question for Tab now. Are allied health professionals who regularly come into residential aged care facilities, for example weekly, eligible to be vaccinated along with the regular staff?
They are. So if you refer to the Allied Health Professionals Australia webpage, and that was updated on the 17th of February, people who are casual, temporary visiting staff, they are actually able to have a vaccination. However, the RACFs have to provide that information along with their list of people to be vaccinated to be counted in the vaccination count. So you would need to talk to your facility and make sure that you're on the list, and they will contact you when the time arrives for their vaccination.
Thanks, Tab. A question for Jake now. Is there an approximate date that this is more for Tab. Is there an approximate date that RACFs in Broken Hill will be receiving their vaccine?
So the government has released a, I guess, rollout plan for that, and it's due to start this week. In saying that, it has changed a few times. So if you want to get in touch with us, we can give you a bit of a general idea, but don't hold us to it, because we don't make the decisions for this, and it's a moving feast. So give us a call and we can have a look at what sort of time frame you should be looking at.
A question from Joanne. Sorry there. Can you clarify for this area /PHN, how many GP practices applied through the EOI and how many have been approved?
I'm not privy to the information about applications, but I know that we have about 55 sites approved, and that's right across to LHD, so that's a fair covering of ground. That information will become available in the coming weeks. It's really just a matter of letting those practices decide that they're on board and that's the way forward for them, because we need to make that information available to the community so they know where they're going to be going for vaccinations.
A question for Jake from Irene. I have a patient who is 16 years old with a spinal cord lesion and is very keen to have COVID vaccination. What's your take on this?
So I guess the question will be around eligibility, right? So I don't hear anything from that patient's specific background that you've mentioned that would say that they have a contraindication. Again, there's a separation between what patient cohort's enrolled in the studies and what eventually the regulator will approve. So we have to wait for that. But I think in terms of vaccinating a 16-year-old with complex medical condition, I would say that would be, when they reach the eligibility thresholds, then I wouldn't see a medical contraindication.
Question from Marianne: how long should someone wait to fall pregnant after receiving COVID-19 vaccine?
Oh, okay. That's a good question. So I guess again, it comes down, if the regulator gets to a stage where they'd be comfortable to approve it in pregnant women, it's more of a moot point. I mean, I would say it's once your vaccine schedule is complete, then I wouldn't see why there'd be any reason not to get rolling. Obviously fever is always something you want to avoid when in early pregnancy or pregnancy generally. So I think the reasonable approach would be, if you've got a vaccine coming up and something that you're considering, I'd wait till your schedule's finished, and then from there you should be right as rain.
A question from Pradeep: what about allergies to flu vaccines?
Yep, so really important. So flu vaccines and COVID vaccines will contain different components. So the nature of the vaccines is different. So most of our flu vaccines are typically attenuated vaccines or inactivated vaccines or recombinant vaccines. So really different platforms for the most part to what we use here. So the two are not analogous, but certainly if you've had a reaction to a flu vaccine, then I would check the product information before providing any COVID vaccine. But I'd say it should be fine.
A question from Sarah: is there anything to substantiate claims that COVID vaccine might affect long-term fertility, or that the vaccine contains components which may damage the placenta in pregnant women?
No. I'm not sure how anyone could infer the long-term effect of COVID vaccines on fertility. There is no safety signal that I have seen, and I've trawled through this data in preparation for this meeting. There is no safety signal of that kind.
A question from Nadine: a frequent question from patients in general practice is, what about the long-term side effects? There's no long-term data. Can you suggest a suitable reply?
Yeah, what I would say is that we don't equally know about the long-term side effects of having COVID. That'd be my first thing. Second thing is, there are lots of vaccines that have been in circulation for a period of time that wouldn't allow us to demonstrate their long-term efficacy. So, what is long-term? If we were vaccinating, you'd have to have a vaccine that's over 50 years old for us to really have a concept of what a long-term safety profile of any of the vaccines we have been applying to the population for many, many years have. There's never been a safety signal for any of the currently marketable vaccines. There's no reason from first principles or their mechanism that they should have a long-term side effect. There's no theoretical concern. There's no practical evidence to suggest it. I can appreciate that people identify that as an unknown, and that can be a source of skepticism, but I think we've got to take a scientific approach here and say, this is absolutely the best approach to take, because things like the meningococcal vaccine are not that old, but no one blinks an eye about giving them all the time. Unless you're a vaccine skeptic generally, I don't see why the COVID vaccine would be any different.
A question.
Engaging with vaccine skeptics generally is a whole different kettle of fish, and World Health Organization has a lot of great resources around that. It's actually very complex, and I'd set your bar very low. Most people who have vaccine skeptics well and truly don't respond to conversations with medical practitioners at all.
A question from Nabila: is there a need for separating annual flu vaccinations with the COVID vaccines?
Oh, that is in the product information. There is a suggestion that you separate them by a period of time, I think a couple of weeks. I can't remember off the top of my head. You'd have to check the licensing of your original vaccine. The product information absolutely will provide that. There is some spacing, but it's mostly just to iron out what an adverse effect might be. Because you can imagine, as Tab said, we've got to be right onto our adverse effect reporting. We don't want to mix vaccines together so that we miss a signal. But I can't provide the detail, I'm sorry.
This is a question from Anna. I'm not sure whether Tab or Jake's going to be best able to answer it. Are pregnant women eligible to take the Pfizer vaccine?
Yes, they are. And so the Pfizer vaccine, I haven't checked Australia's licensing requirements specifically, but certainly the Pfizer vaccine was given to 1,000 women in that Israeli study, as I mentioned, without a significant safety signal. Most of its product approvals overseas have included pregnant women because it would relate to, there would be too much interrelation in the rollout. I can't quite remember what the TGA said. I'm pretty sure there wasn't a restriction. So even though it has a relatively high rate of fever, there has not been advice not to provide it to pregnant women.
Another question from Mrna. How long should a patient wait in the pharmacy to ensure there's no anaphylactic shock?
Generally speaking, we ask people to wait. When we're doing them in hospital, we ask people to wait for 20 to 30 minutes. We have a specific waiting bay set aside because the Pfizer vaccine is a multi-dose vial, and this might be less relevant, I guess, for the community settings where you'll use a different vaccine candidate. Everyone would have seen the unfortunate incident that happened in Brisbane. The reason that happened is because these vials, you draw up between 4 and 6 doses out of each vial, and traditionally it's 1 dose, 1 vial, boom, and that was where the mistake was made. And so because we've got to immunize a whole batch of people at once, basically using this vial, we've got a designated waiting space. But that would be specific to the Pfizer vaccine because of its formulation.
Okay. If there's any more questions, we might have another minute to answer one.
And just back to the spacing between vaccines, again, that's a moving feast as well. So the recommendations do change on that as time goes on. So the AstraZeneca one at the moment sits at 4 to 12 weeks, and ATAGI in Australia is saying 12 weeks. So you just need to keep reading the evidence, because as they're giving vaccinations and seeing the different impacts of the spacing between the 2 doses, that may change. So just refer to your product information, but keep your eye on the evidence as well.
Yeah, that's right. I mean, what I've done in preparation for this talk is go through the studies, but there's this overlay of regulatory frameworks. So essentially, it's very difficult for me to be a direct line from telling you what the evidence says is safe to saying what you should do in your patient populations, when really it's ATAGI, the regulator, and the governments who provide the vaccines who will tell you how and when to use them in certain populations. But I can give you a general impression as to what the data says.
We do have one more from Nabila. How do we manage safety of administering vaccines in a pharmacy setting if there's multiple dosings?
Yeah, so that's specific to the Pfizer vaccine, and as Tab was pointing out, the Pfizer vaccine probably won't be part of the rollout by the time community pharmacies are immunising. So it should be less of a concern for you. It's certainly something we're having to provide specific training for Pfizer, but I wouldn't worry about needing to use multi-dose vials in the pharmacy setting for that reason. You should just be able to use the AstraZeneca vaccine, which is single dose, single vial.
What about vaccinating over 70s with blood cancers?
Yep, so that is something that for other vaccines has been a thing issue in the past. So the shingles vaccine comes to mind. Again, we'll have to wait until the regulator makes a specific statement about those patients, because it will come as part of that rollout population. But from the available data, there is no concern because the concern, I guess, in those populations is often if you're giving a live attenuated virus with a weakened immune system, you can cause functional disease. But because of the structure of the the vaccines we're using, it would be an efficacy question, not a safety question. Will their blood cancer prevent them from generating an appropriate immune response to make it effective? Not will it be unsafe? So I think it's we'll have to wait until that detail is clarified, but I don't foresee a large safety issue.
That's it for the questions, so we might finish it there. Oh, I'm sorry, we've got one more from Sarah. Any news on when children will be added to the rollout?
Very, very end, I reckon. That's my personal view. All opinions my own. I reckon the very, very end. Because there's a few reasons why that is. Children are very hard to recruit into clinical trials because the consent issue and obviously the concern around safety. It's the same reason why it's harder to study pregnant women. And it's also, felt that their chances of severe disease and onward transmission are variable, but probably on a lower scale. So I think they're likely to be well down the end of the vaccine rollout.
And just quickly, just from my brain's trust on the phone, they've just let me know that the prioritisation for RACS is Orange, Bathurst, and then Dubbo, and then it'll be Wellington, Yeovil, and then Broken Hill, for the Broken Hill people that asked. But we can't give you a time around that because that's a moving feast. And just to adverse reactions, the public health unit does not want to know about minor reactions like pain at the site. They only want the serious stuff and the stuff that needs reporting. So just also be aware of the things that are reportable to save yourself time and the public health unit time about going through all the reports that have come through. Thank you, guys.
Thank you, everyone. There's an evaluation QR code on your screen there. If you could do an evaluation survey, that would be great. We just have a comment from Michelle. Sorry, both current vaccines are multi-dose. Flu vaccine can't be given 14 days either side of the COVID vaccine. Thanks, Michelle. Okay. Thank you, everyone, for coming tonight. And if you could do an evaluation survey before you go, that would be great. Thank you, Jake. Thank you, Tab.
Thank you for your time, guys.